Hello everyone, welcome to Get My Class. So let's solve this question which says nematode-specific genes, that is DNA, were introduced by using Agrobacterium vectors that produces option A, both sense and antisense RNA in the host cell. Option B, only sense RNA in the pathogen cells. Option C, only antisense RNA in the host cells. Option D, both sense and antisense RNA in the pathogen cells. Now let us understand the question. It says the nematode-specific genes were introduced by using Agrobacterium vectors that produce. So here they're talking about the pest resistance, production of pest resistant plants. So they are introducing this nematode-specific genes to produce pest resistant plants. Okay, pest resistance plants. Okay, plants. So when we want to produce pest resistant plants, in which organism are we supposed to introduce the gene for the resistance? That is, of course, in the plants. Okay, that is, plants are nothing but the host here. And the nematode is nothing but the parasite. The plant is the host. And nematode is the parasite. Now we are going to make this plant or the host as resistant towards this parasite. For that, what do we do? But how does this parasite is affecting the host plant? By producing toxic proteins. It will produce toxic proteins, which affects the yield of the plant. Okay, now this toxic protein to be stopped. We don't want the toxic protein to be produced. So what do? We introduce the gene for that particular protein in the host by Agrobacterium, okay, which will have the Ti plasmid. The Agrobacterium will have Ti plasmid, and we will, we introduce in the Agrobacterium, and further we allow it to infect the host plant. By what? By Agrobacterium. Then Agrobacterium will release its Ti plasmid within the host cell, that is plant cell. And this Ti plasmid carries the gene, okay, for this particular protein. So what will it do? When this nematode parasitize these cells of the host, okay, suppose the nematode is here, it will produce its own RNA, right? And it will form the protein, right? But here, this host containing this Ti plasmid, containing our gene of interest, it will produce, I'll just show you. It will produce two strands, that is sense strand and antisense strand of the mRNA for this particular protein. Okay, this is sense strand and antisense strand, because double strand DNA, right? Double strand DNA, and each DNA will act as template for transcription, that we all know. So if one strand is acting as a template for sense strand, the other strand will be acting as a template for the antisense strand. So both the sense and antisense strands of mRNA will be produced where? Within the host cell. Now in this, so whichever the RNA is complementary with the RNA of the parasite, it will bind with that and convert it into double-stranded RNA. So let me show you. So suppose this is sense strand and this is antisense strand of the host. From the host it is coming. From the vector. Okay, from vector. Okay, this is forming within the host. Okay. Now here is your you can say nematode, and it is the parasite, and it has formed its mRNA. Okay, and now what will happen? The mRNA, whichever is complementary to this particular RNA, it will come and bind to it. Okay, so let me show it in the other color. Okay, suppose, as I told you, suppose this is the host, and we have introduced the vector, and this vector has given rise to both sense and antisense mRNA. Okay, both are mRNA. Okay, but both are single stranded, and one is complementary, like both are complementary to each other. Okay, sense and antisense. Now you have the nematode here, for example. Nematode is here, and it has produced its what? The RNA for that particular protein. So of course, This also has its gene. This also has its double-stranded DNA. This also will produce sense and antisense RNA because this also double-strand. What will happen? The complementary mRNA from the vector what we have introduced inside the host will combine with the complementary mRNA with respect to the nematode mRNA. Okay, so this will happen like this. So this also getting converted to double-stranded RNA. This also is getting converted to double-stranded RNA. That means we are not allowing even a single strand of the nematode RNA to be translated. Okay, so because it is getting converted to double-stranded, it will not get translated. So no protein will be formed. Okay, so the expression is happening where? Within the host cell. Okay, so let us see. Option A says both sense and antisense RNA in the host cell. Yes, that is absolutely right. Option B says only sense RNA in the pathogen cells. No, because we are introducing the gene in the host cell. Okay, so it has to produce in the host cell itself. Option C says only antisense RNA in the host cell. No, because we are introducing double-stranded DNA. See, this vector which is we are using is a double-stranded DNA vector, right? And of course, it will code for sense and antisense RNA. Then option D says both sense and antisense RNA in the pathogen cells. So we are introducing the gene, and that will definitely we are introducing the host cell. So we have to talk about the sense and antisense RNA in the host cell. Hence it is ruled out. So what is the correct answer? The correct answer is option. Let me write here. The correct answer is option A. I hope you understood the explanation. Thank you.